Well, it's a pleasure to be here again, and I'm very happy that Richard and the crew was able to invite another relic, because I'm certainly in that category. And particularly to talk a little different vein this year. And, you know, I was struck that in spite of 25 years now of a lot of basic research and a lot of clinical research, that we are still struggling with some basic issues of etiology, diagnostic criteria, pathogenesis, treatment, et cetera. So we must be close to setting records in an unknown disease. And you know, you're probably all aware that there's about 6,000 published articles now. So that's really a lot of literature describing things kind of around the bush or parts of the elephant or however you want to describe it. I think there's a lot of explanations for this lack of success, but very few excuses. So I'll tell you what I think the two excuses are. The greatest challenge probably is the disease itself, being so heterogeneous in onset, diagnostic markers, symptoms, multi-system problems, and then the lack of a good reproducible biomarker, which we have all been looking for a long time. And that is a real challenge for basic researchers, but I think it's an even bigger challenge for those trying to do clinical research. And I was very inspired by the Norwegian journalist last evening that was able to so articulately manipulate words to an optimistic conclusion that maybe we've turned the corner and that there's light at the end of the tunnel. And I think you certainly saw a lot of that quality of basic research this morning. And I just wanted to bring you up to date on some aspects of clinical research that are going on, at least in the United States, in the past 12 months. And a lot of this is perhaps the silver lining of a very black cloud that occurred in clinical medicine in the last year or so in the US with respect to this. But we also have new tools that were not available to us, certainly when I started in the field. And some of that is translational medicine, the resources that concept brings to clinical research. And there also has been the development of informational technology that we certainly did not have until maybe the past 5 or 10 years. And these are really come to bear in clinical trials, not just in CFSME, but in many, many areas of medicine. So I think you all know Francis Collins, our friend and director of National Institutes of Health. He does direct a lot of directions within the spending of healthcare dollars in the United States. He was formerly director of the National Human Genome Research Institute, and is a very well-respected, famous physician in the United States. And as you may know, he's a great proponent of translational medicine. And he speaks to it very broadly, and he does acknowledge that there's a lot of problems with the institution of the concept that theoretically is quite straightforward. I want to take an example of an NIH program. Progeria affects 1 in 8 million children, for a total incidence of 100 in the past century. CFS affects 400 per 100,000, yet NIH has diagnosed the genetic defect and is working on a treatment trial to remedy this problem that I think you would all agree is minor in number, although also severe. The NIH, as you know, as our leading research institute, has spent very little interest and even less money on this problem. There are principles of translational research that I wanted to go over quickly with you. It's really the concept of integrating basic science with clinical trials. And, you know, for years we've talked about this concept of bench to bedside. But again, it's very difficult to navigate those waters. The advantages of it are, it is multidisciplinary. We can take all the kind of people that presented today and put it together with people who see real patients, or pharmaceutical companies, or any number of combinations that we want to utilize. It does add accountability and standards, particularly to clinical medicine. And we have been guilty of doing N of 1 studies and poorly controlled studies and poorly funded studies and poorly completed studies and anecdotal studies and poor publications. And this change is a very welcome one to clinical medicine. It does allow shared data and data integration, and that's very important these days. And I will point out that you do have to have a common goal and a common drive toward advancement of some kind of applied science. In other words, you can't just have a basic researcher who's interested in some point of NK cell function unless it's somehow connected to a disease process that has an end. And that's driven by many economic factors. There's some basic advantages that everybody recognizes now. One is that you can have larger sample sizes. You can certainly get a more unique and properly categorized patient group. You can have increased number of controls. You can theoretically decrease the cost of research. And you can have increased value of the results to health outcomes. And that's very important in the United States right now. That whatever you're doing, it has to have some outcome measurement in terms of costs and benefit to society and payers. There's certainly decreased timelines, and I'll show you examples of that. And it's much more manageable for non-full-time research physicians and active clinicians that don't have the resources to research CFSME full-time. There are also some disadvantages. Technology is definitely playing catch-up. We physicians have all had problems with our web-based electronic medical records. In a lot of hospitals, they just plain don't work. There's a new level of operational management that's really required in order to manage these huge amounts of data cohorts, biobanks, et cetera, that are being established. And as I mentioned, government agencies and pharmaceutical companies feel they've spent billions of dollars on basic research that has not led them to a marketable product. Many patients and physicians are very apprehensive about this style of data collection, and I'll go over some specific examples about that. And there are some possible security risks and technical complications. Another drawback potentially is that it's very easy to collect a huge amount of information, but a lot of this may be irrelevant or peripheral and difficult to statistically rationalize. So I think more is not always better. So I'm going to review in the next few minutes the XMRV study, the novel pathogen study, the collaborative spinal fluid study, the Bond University study that you've already heard about, the very large CASA project, the CFIDS of America grant awards, and lastly, the multi-site clinical assessment of chronic fatigue syndrome sponsored by the Center for Disease Control. These are all new projects in the last year, so I think it really demonstrates to you the changes that have occurred. You're all probably very interested in the XMRV and MLV and chronic fatigue study. As you know, it was funded by the NIH on an intramural grant. They chose 5 geographically unique clinical sites in order to be sure that if there was an agent involved, that it wasn't unique to one part of the US. 25 cases, 25 controls at each clinical location. All the cases met the Fukuda and the Canadian consensus criteria, and the healthy controls were carefully matched by age, gender, ethnicity, and geographical location. And one of the advantages of this kind of study is you can have a large number of controls that are built into the funding of the study. All data instruments were included for both the cases and the controls that were treated identically, and there were exclusionary criteria that you're all familiar with. Questionnaires were completed by both the participants on paper in this case. A centralized lab processed all the lab tests so there could not be regional variation. The required labs were drawn at the same time as the research blood draw. They were shipped in the same container to avoid any allegations about containers or shipping problems. The data was collected very broadly with demographics, questionnaires about symptoms, past medical history, surgical history, validated fatigue scales, et cetera, and physical examinations were completed by the PI at each of the clinical location and also completed on the controls. Each participant was pre-screened over the phone by the study coordinator, and then about a 1.5 hour visit for the patients and a half hour for controls. The difference is there that the controls can answer the questionnaire much more quickly than the patients. The blood draw and physical exam was then completed in about a half an hour, and cases and controls were both reimbursed for their effort. The samples were sent by Federal Express overnight to the Center for Infection and Immunity at Columbia University, and Columbia handled the CFFs for the project. Now, the advantages of this is it was a pretty clean and simple and effective study design, and it had very tight and very well enforced timelines. This allowed for very rapid enrollment of cases and controls, and there was a very effective leadership from the CII with respect to quality control throughout the process. It was very easy to recruit for. There was great demand to be included in this particular study. The drawbacks, the questionnaires were stressful for some of the patients because of their cognitive dysfunction, and it was a relatively low sample size that was calculated to show statistical significance based on other reports in the literature. The laboratory analysis included 3 sites, the CDC in Atlanta, the FDA in Washington, D.C., and the Judy Mikovits lab in New York City. The sites did different techniques. They did PCR of plasma at the CDC, PCR of PBMCs at the FDA, and the Mikovits well-published culture and serology technique that you're probably all familiar with. I'm really happy to say this study's been completed. The results are in, and the statistical analysis has been completed, and this will be released on June 30th, 2012. In addition to the results of that particular study, we have established a really well-characterized cohort of patients and controls at 5 sites in the United States, and the CII is very open to inquiries of utilizing this database and their collected biorepository, although it was quite small in size, the repository I'm speaking of. The next study that you've all heard about, I'm sure, is cumbersome in name, a clinical and biosample database to enable discovery of pathogens and pathogenic mechanisms in chronic fatigue syndrome. We call that the CFI Pathogen Discovery Project. This was sequential to the XMRV study for the clinical sites. Its goal was to create a well-defined biobank and database that's accessible to researchers. The funding institution was the Chronic Fatigue Initiative, which is private funding. The major addition was the use of a RedCap web-based data collection tool, and it was designed for ease of clinical research studies. Again, it also has advantages that the information can be exported to a number of statistical programs and utilized by those interested. Here's an example of a RedCap data entry sheet, and this can be emailed to the patient so they can complete it in their own time frame. You can restrict access to various people participating in the study, and you have access to some raw data very early on in a trial such as this. So for this particular study, 5 clinical sites were required to recruit 40 cases and 40 healthy controls. Again, a very large number of controls for a study like this, and there were statistical reasons for that. The controls were very closely matched to the cases. It required tissue collections, and the cases and controls, again, were required to answer very extensive demographical questionnaires with respect to medical history, past and current symptoms, medication history, et cetera, and these were all entered into the RedCap database system. A little difference in this particular study, each site was required to recruit 10 acute subjects, and the reason for that was if you're looking for a pathogen, either known or novel, it was felt that it would be more likely to be detected in somebody who was acutely ill versus somebody who had been very chronically ill. And then the remainder were more classical long-duration chronic fatigue patients. In addition to the questionnaires and sample collection, the coordinators did extensive chart reviews on each patient, and there was a unique system utilized for that I'll come to in a minute. The cases and controls also donated, in addition to serum and plasma, urine, rectal swab, tears, and saliva, allowing very extensive evaluation of other tissues in these patients, because the question has always arisen, whether or not the peripheral blood is actually the place to look, particularly for a pathogen. The coordinators were required to do chart review. It was very extensive, and the technique utilized for that was the point A was when the person was first seen, and extensive data was obtained from what their illness was looking like when they first presented, and then intercurrent events were outlined, including any surgeries, hospitalizations, et cetera. Then the last visit with the patient was recorded, and those were contrasted. In addition to that, the PIs drew a trajectory of the illness that was then coded into the database. And the reason for that is we all know when we're involved in the clinical history here that many of these patients started with an acute flu-like illness and then have developed what you might describe as a chronic neurological disease. And we wanted to grasp that trajectory rather than just one point in time, and this was one way to do that. So the labs that were required for both the cases and the controls. They had to be completed on the same day at a set time. There was a very close time window for this, partly because of looking at cytokines and things like that. Coordinators also had to extract any labs that had been done on any patient over this entire duration, such as NK cell function, cytokine profiles, viral studies, imaging reports, exercise tolerance tested, sleep studies, tilt table tests, et cetera, et cetera, et cetera. This became the very time-consumer and labor-intensive part of this particular study. All of the information was then scanned, redacted, and uploaded to RedCap. Here's the screening labs that were done on both the patients and the controls, basically for exclusionary purposes. There were some problems with this technique that RedCap does have some technical barriers, making it a somewhat difficult system. The cases and controls could, however, do it at an off-site location and at their convenience, and that they rather liked. It does take really skilled and dedicated individuals to complete a clinical platform like this. Many of the patients preferred to use a pencil and paper. They simply did not like this web-based online approach. The controls complained that a very long time was required to participate in this study. RedCap is overall useful, but it has some faults that probably will be straightened out in the future. One of them is that it's a free program and lacks IT support. It is, however, I think, suited for this kind of project. A couple of lessons that I've learned is to do this kind of clinical research, you really require a staff that is well-trained and have constant vigilance for quality control. The CFI has, in fact, addressed these issues. They have face-to-face coordinated clinician meetings every single week. The coordinators participate in weekly meetings with respect to enrollment, RedCap issues, and general day-to-day updates. And the electronic platform, once you get used to it, really does increase efficiency. The goals, of course, is to identify specific subsets of CFS patients with respect to pathogen discovery. The CFI model allows for collaboration with many clinicians who could not otherwise have participated in such a major, expensive, and extensive study. There is a centralized database and biobank being established for future research. That study is about 60% enrolled now, so I would hope that by next year at this time, we would have some information to report. The collaborative research using cerebral spinal fluid study is also funded by the CFI as well as the Edward P. Evans Foundation. The collaborators are Sierra Internal Medicine, Wisconsin Viral Research, and the Center for Infection and Immunity. The goal of this study is to identify aberrations in the spinal fluid of these patients and compare those to MS controls. And this is established from a large CSF biobank of over 400 specimens collected over 25 years that have simultaneous serums, PBMCs, and in many cases longitudinal spinal fluids. These have been maintained at -80 and in most cases have matched specimens. 30 cases are selected from those who are already enrolled in the CFI pathogen discovery, and the reason for that is to correlate findings in the peripherally with those in the spinal fluid, and they again meet the criteria. 30 other cases were selected based on their neurocognitive complaints. And disease-positive controls were those with other neurological inflammatory diseases, 18 normal controls, and 33 patients with multiple sclerosis. Again, the pathogen arrays are available at the CII at Columbia University for this study. And again, an attempt is to differentiate subsets of populations, perhaps, and to provide other pertinent findings to other collaborative researchers in the world who are interested in the findings in the spinal fluid. You've already heard quite a bit about this study, the molecular and cellular investigation to identify NK dysfunction, a joint project between Sierra Research and Bond University. And again, the goals, as you've already heard, were to look for the mechanisms of reduced NK cell lysis, look at the abnormal microRNA, the cytokine profiles, again in the spinal fluid, and then to look at the purinergic mediators that we've heard a lot about today. That is also in process, and I'm happy to say that the pilot study has been completed, and we're waiting for the data analysis on that. Many of you know that the CFIDS Association of America has been committed to funding research projects, generally small projects, over the past 20 years or so, and they have endorsed the translational model. Their new program, which you may or may not be familiar with, is called Research Institute Without Walls, and it's aimed at identifying clinicians who can contribute in their own personal way according to their interests and without spending money on buildings or other infrastructure. They committed to funding 5 studies this year, and those are in the process of being executed at the present time. I'll go over those very quickly to give you a flavor of what other sites are doing. Dane Cook, who many of you know, has been active in this field for many years, and he plans to use brain imaging and blood markers with respect to exercise. And this is really a continuation of the LIGHTS work, where exercise-induced upregulation of gene expression. Most of you are familiar with this. It's kind of an extension of that project. This is new, and this is a goal of the NIH, to look at drug repurposing opportunities. And this is not just for chronic fatigue syndrome. This is a concept that's being used in a lot of diseases because drug development is so expensive. And what BioVista Corporation had proposed is that cognitive impairment and non-refreshing sleep are very disabling symptoms for the patients. And there may be in the armamentarium of already licensed drugs agents that are extremely useful that wouldn't require extensive phase 1, 2, and 3 trials and could be short-circuited, much to the benefit of the patient. This is a very interesting pilot study, I think. You're all familiar with this burgeoning field of epigenetics, and Patrick McGowan at University of Toronto was funded to look at epigenetic alterations of the signaling pathways, and he is going to utilize the biobank, the Solve CFS biobank that was established by the CFIDS Association of America. And Marvin Meadow is looking at brain fog and CSF with respect to reduced cerebral blood flow, impairing cognition with respect to orthostatic challenges. And what's a little different about this proposal is that he's proposed to do some therapeutic interventions, such as rebreathing of CO2, IV phenylephrine, and IV acetazolamide, which are agents that are known to increase cerebral blood flow. And lastly, Peter Rowe, who's another well-known researcher in this field with an interest in autonomic dysfunction, has been funded to look at neuromuscular strain as a source of sensitizing input. And we really heard a lot of that basic science today, and he is trying to apply this on a clinical basis. The collection, aggregation, storage, and analysis of ME-CFS, or the CASA project, was established by the CDC. And the purpose was to continue ongoing discussions of the most efficient, systematic, and standardized way to collect aggregate, store, and analyze information from these patients. This is felt to be long overdue in this field. If you read the literature, you'll see that everybody uses their questionnaire or their fatigue score or their modified physical examination. And it's very, very hard to compare studies, which really oftentimes holds up progress. And the goal is, of course, to ultimately publish this consensus document as a guideline to researchers and clinicians who want to do research in this field based on the translational approach. This has consisted of bi-monthly calls of clinicians, researchers, private citizens, government officials, IT experts, consultants, et cetera, studying different domains. And the domains that have been done already were pain, fatigue, sleep, neurocognitive, autoimmune, neuroendocrine, immune, functional studies, and mood and global well-being. It's sometimes difficult for a mixed group, such as I'll show you here, to agree on anything, much less the best tools. But we're giving it a shot, much as Bruce Crothers did in the 2003 definition, and I was always impressed by that collaborative consensus type of approach. Here's just an example of 4 chronic fatigue ME studies that utilized entirely different questionnaires and procedures, and you can see why that would be very difficult to compare outcomes from such a collage of collection techniques. And the hope is ultimately publish those guidelines so that we can use the best techniques that are out there. And lastly, the multi-site clinical assessment of chronic fatigue syndrome project, which is a product of the National Center for Emerging and Zoonotic Infectious Diseases, funded by the Center for Disease Control and Prevention in Atlanta. This contract was awarded to the Open Medicine Institute, as well as Dr. Nancy Klimas at Nova University, and Dr. Ben Nadelson at Beth Israel. So it's multi-center. And again, the sites are responsible for recruiting at least 75 patients with validated questionnaires and fatigue scales. And each clinical site will use the Open Medicine Institute electronic medical records platform throughout the study. And we'll have the option to continue using this software in our research projects. The Dr. Kogelnick from Open Medicine Institute, this has been one of his, I think, dreams and hopes to get this project off the ground. Again, I'll allow him to speak about it, but it really, again, tries to marry clinical medicine and research in a really effective fashion and add those deficiencies we had of informatics, biosampling, and social networking in the concept of a collaborative translational research institute. And just briefly, and he'll talk more about this, the patient has portals. They can enter in all their own information and access it. The provider can access it through the electronic medical record. The lab is automatically inputted into the system. The researcher can access de-identified research data, and pharmaceutical industry, et cetera, can access the same information, the same patients through clinical trials, and there's a large biobank being established. So in conclusion, I think there may in fact be light at the end of this long tunnel, even with respect to clinical trials and investigations. Thank you very much.